Hello, everyone. Welcome to today's live broadcast, Squeaky Clean, Direct Spectroscopic Measurement for Cleaning Validation. I'm Laura Bush, the Editorial Director of Spectroscopy, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Spectroscopy and sponsored by Helma Analytics in collaboration with Prozess Technologie. Helma Analytics is a world leader in the market of optical analysis technology for optical components and solutions in glass and quartz glass. Their high-quality precision solutions provide the basis for achieving reliable measurement results in production, quality assurance, and research. Thanks to their outstanding performance level and consulting expertise, Helma Analytics is a favored partner for manufacturers and end users around the world. Since Helma GmbH was founded in 1922, their commitment has been to provide the best possible quality to guarantee the most precise analytical results. Prozess Technologie is a technology company that designs, builds, and implements spectroscopic solutions for process measurement and control in multiple manufacturing sectors. The company uses precision light technologies to measure, enabling customers to understand and control manufacturing chemical processes in real time. The company's integrated hardware and software solutions are in use by leading global manufacturers in the life sciences, food and beverage, and petrochemical industries. Prozess Technologie is a privately held company based in St. Louis, Missouri. We have a few housekeeping announcements before we get started. The webcast is designed to be interactive, so we encourage you to ask questions. You can submit questions at any time by typing them in the Q&A box, and you can find that on the right-hand side of the presentation window. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically. If you have any technical problems viewing or hearing the presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. Please also note the valuable resources available on the left-hand side of your screen, provided by our sponsors. I would now like to introduce today's speakers. Chad Lieber has been at the forefront of developing advanced spectral instruments, including UV-Vis, NIR, SWIR, and Raman analyzers, spectral imagers, and Raman microscopes for the biomedical, defense, pharmaceutical, food, and petrochemical industries. He joined Prozess in 2013 and leads the R&D and engineering efforts for its portfolio of process analyzers. Before joining Prozess, Lieber was a senior application scientist at BaySpec, coordinating custom spectral system design for customer applications. He was the founder and principal investigator of the Bio-Optics Laboratory at the Children's Hospital of Orange County in California, and concurrently a research professor at the University of California Irvine's Beckman Laser Institute, developing optical diagnostics for biomedical applications. Earlier in his career, he was a senior scientist in the Johnson & Johnson family of companies, leading efforts to integrate optical technologies into cardiac catheters. He holds a PhD from Vanderbilt University and performed postdoctoral training at the University of California Davis Center for Biophotonics. Evan Friedman has spent nearly 10 years in the field of spectroscopy and analytical fiber optic devices with Helma Analytics. He is a resident expert in fiber optic connected spectroscopic sampling devices. Currently, Friedman oversees all the sales and marketing operations for the Americas region. In this role, he is responsible for client account management, business development, technical service, and distribution throughout the region. Friedman studied physics and optical engineering at the University of Arizona, as well as business at SUNY Farmingdale. Thank you both for joining us today. Chad, please go ahead and get us started.
Thank you very much, Laura. So today, as you all know, we were going to be talking about cleaning validation using a direct UV spectroscopic approach. So what is cleaning validation? It's one of the behind-the-scenes features of any manufacturing process, particularly a batch manufacturing process, that's critical to avoid cross-contamination between batches. And specifically, as we're talking today, we're talking about cleaning in place or bringing the dishwasher to the dishes. as we'll get into in the details on the following slides, to ensure the cleanliness and sterility of manufactured products. So first, we're going to talk about the status quo of what's currently being used outside of direct UV spectroscopic methods. And the challenges are that stopping cleaning processes for lab tests or for swab tests is inefficient. And furthermore, that indirect methods that are not specific are also less robust. There's a particular importance right now on water conservation, given a lot of the droughts and water shortages in a variety of regions of the world. That by having a real-time view of the chemistry that's being undertaken in your cleaning process is perhaps more important now than ever. So while current methods such as swab testing or offline HPLC methods are very accurate, they're also quite slow and costly, both in terms of the time taken away from the process as well as the monetary cost to keep these methods running continuously. Indirect measurements such as things like conductivity are less robust and not as specific. So for these reasons, this is why we're talking about improving efficiencies in manufacturing and in cleaning validation. As I said, everyone now, if everyone looking at a newspaper or any news source is aware of the persistence of drought. And in some of these markets, the penalties for overuse of water is considerably more than implementing real-time solutions. This is not just an American problem. I'll point out this is a worldwide problem. So today we're talking about a direct UV spectroscopic application as part of a broader PAT approach. And as I'm sure most of the listeners are aware, there are a variety of benefits of an inline PAT approach that adhering to a QBD approach that has been developed in a rugged and robust way provides a lot of benefit. Basically, taking the lab and putting it into the manufacturing process can be used continuously or in batch. It can improve the process, can improve efficiencies, can eliminate waste, can eliminate risk. So we're going to talk a little bit about our solution, and I'm going to start talking about the process aspect of this solution before Evan picks up and talks a little bit about Helma's contribution to the solution as well. So we're talking about UV spectroscopy today. While there are a number of optical spectroscopies that can be and have been applied for cleaning or a variety of other process approaches, UV is well suited. Near infrared can be used, but the detection limits are relatively high compared to UV. There you're talking about percent level or fractional percent, which typically will not allow you to validate a cleaning process. Methods like IR can be used as well. There is a difficulty in using IR in aqueous environments. Raman and LIF can also be employed. In some cases, Raman does carry along some technical. The increased sensitivity and specificity of Raman does come at a technical expense. And methods like fluorescence or light-induced fluorescence can also be used, but then depend on your samples to be fluorescent. The great benefit of UV approaches is that nearly all pharmaceutical APIs or excipients exhibit some sort of UV absorption features, whereas a lot of the cleaning solvents have minimal or no UV features. Further, UV spectroscopy can also reduce your limit of detection considerably down into a low parts per million range, which is typically adequate for demonstrating cleanliness. So as I had said earlier, there are some challenges in the current approaches to cleaning validation, and everyone seems to approach cleaning validation from a different standpoint. But I think from a common common viewpoint, the challenges of cleaning validation are determining the proper endpoint, and doing this particularly in an at-line or offline approach where you're grabbing samples, taking them to a lab, is very inefficient and can add hours or days to a process. Also, one of the problems is that these cleaning recipes that are developed in formulation or in scale up in pilot don't necessarily translate into production batches. So typically the cleaning protocols will entail an excessive rinse and wash cycle that is not always necessary. So the solutions that we'll be talking about are talking about real-time monitoring in the effluent in the drain line, where we can use this approach both qualitatively and quantitatively to determine endpoint, so that you can enable a closed loop integration, so that the manufacturing efficiency can be most improved. This is a quick cartoon of how, or I should say a diagram of how this cleaning validation actually works. So if this is our apparatus here, the gray box installed on one of the drain lines, we'll can send the analyzer control, wired or wirelessly, depending on where the unit is, to control the spectroscopic aspects of it, how quickly it's making a measurement, integration times, the wavelength ranges that are going to be used, and so forth. And from that, a spectrum is acquired, and in a lot of cases, the spectrum is not the endpoint. They're converting that spectrum into the constituent monitor, which is typically the most dangerous compound in a formulation, or all compounds in a formulation. To get to that, the spectra are pre-processed. The pre-processed spectra are then sent into a quantitative model, so that now the constituent or the amount of remaining residual API can actually be monitored in a quantitative sense, in a parts per million approach that can then be reported upstream to control systems for that closed loop integration. And I'll show some data in a few slides to actually show this in a little bit better detail. So how does the Prozess solution work? This is an example installation of a Prozess analyzer about the size of a shoebox, installed in some sanitary tubing. You can see that typically a cross-flow cell or a transflectance probe will be used, and Evan will certainly talk about that in greater detail than I. But to monitor this fluid, and we can look, as I had said before, many of these cleaning solvents are UV transparent within a certain region. So that allows us to look through the solvents at the APIs or the rinsate directly. If you do go far enough down into the UV, you can also look at these cleaning solutions as well. So if it's important that you conserve detergent, or if you're conserving any of these cleaning solutions, you can monitor those as well. And the idea is that you're measuring either the saturation of your wash solution with product. So when it's rinsing, you know that your solution is saturated, and doing further rinsing does nothing but just increase your wasted time. so that then you can flush and rinse again until your solution is not saturated or until you can eliminate product. It's also good as your flushing systems where you end up getting clogs or buildup of the contaminants, that as you're flushing you can actually see these release in real time. And one of the further benefits of this approach is that by using one or multiple spectral models, you can cover more than just one range. You could have separate models, for example, to look at a broad range of concentration if you're looking at percent level dilution, and then transition into another model, and these can be running simultaneously on the Prozess systems, so that you could look at trace concentrations as well for that final cleaning validation. This again is a picture of the Prozess solution with all these benefits that we've talked about. Certainly not just in cleaning validation, but in a number of applications. Just a quick look of what's inside this box. Probably not a huge surprise to anybody. So the Prozess solutions are all based on a similar platform with common control electronics and communications. And to that, we'll adapt a spectral engine suitable for an application. So in this case, we're talking about UV spectroscopy. So that's built on the same platform as say, one of our near IR solutions. So these could be adapted very quickly as well. And the application science box, we've shown it as being inside the box for those cases where modeling would be desired by, say, for example, a customer without any in-house PAT, where they don't have the capability to do modeling. We can build some generic models for them, or there are also solutions to do this externally with some control software that I'll talk about shortly. So in addition to having that standard box that we've already shown, Prozess also builds these systems. It can do custom integration as necessary. A lot of the UV cleaning applications are done in hazardous environments. This is an example, this box here that shows the same internals, but now in a Zone 1 approved chassis. We can also apply different communication support or multiplexing as necessary for each requirement. And the sample interface, as I had said before, is typically integrated directly into a drain or a bypass line to look at the effluent. And as I had said before, it's typical use of an immersion probe or a process flow cell, and I'm going to let Evan talk a little bit more about those because they have some exciting new developments on that front.
Thank you, Chad. So as he said, this is typically integrated directly into the drain or bypass line for cleaning validation. Frequently, for other applications, we can integrate directly into continuous flow pipelines and batch reactors as well, using our fiber optic connected immersion probes. For 93 years, Helma has been making precision spectroscopic sampling accessories, and for the last 20 plus years, we have been designing, manufacturing, and advancing online process sampling with fiber optic connected immersion probes. We've developed a modular system to allow us to produce online sampling interfaces for virtually any environment, where we can customize the barrel materials, the diameter of the barrel, the light path, the temperature range up to 300 degrees Celsius, the pressure range up to 750 bar, and resistance to a wide range of solvents from highly acidic to extremely basic. Most recently, this year, we've completed the 3A certification process on our hygienic design probe. What exactly does this mean? Many food and beverage applications are required by the USDA to conform to sanitary standards that have been standardized in the U.S. under the 3A Sanitary Standards Incorporation. In Europe, it's done under the EHEDG, and it means that the sampling interface must be able to be cleaned thoroughly, and it cannot have any areas where sample can get caught and bacteria can grow. This is especially important in food processing, but can be critical in specialty chemical applications where crossover can severely impact batch yields, as one example. So the barrels are electropolished; they have no cavities. The optics are made out of sapphire to ensure that if something were to impact it, it wouldn't break off and end up in your finished food goods. So the typical applications for a probe like this primarily is milk and dairy, but biopharmaceuticals are critical, as well as you have living growth going on and need to ensure cleanliness there. This probe, with its hygienic design, is perfect for CIP procedures because you know that it can withstand the cleaning process and validate that the rest of the system is clean. And we have developed these standards to make it easier for adherence to USDA and FDA regulations. So I'll hand it back to you, Chad, to talk about a case study itself.
Sure, thanks, Evan. So before talking about that, I also wanted to mention that we had talked about it on a previous slide about how this actually works. But in addition to the hardware component provided, the analyzer that Prozess develops, the sample interfaces that Helma develops, Prozess also has control and chemometric software that has been around for now about a dozen years. With that is also critical to this aspect. So not only all the necessary compliances, but it enables the real-time use of these models. As I said, typically up to 12 different constituents that can be tracked simultaneously, and this can be in millisecond time frames. And also the controls to automate the internal calibration procedures and validation procedures for USP. So, talking a little bit about a case study, just to show some data. So this is an example of a cleaning process. And here we're anonymizing the sample. This is a pharmaceutical API that we're measuring the elimination of. And here you can see that in the methanol, developing the calibration data on this towards a quantifiable model. You can see an elimination of the absorption with concentration of the product from this. Before I skip forward, I should also mention the range that typically we'll monitor starting around 190 nanometers. And we have various options for the longer wavelength and depending on the necessary resolution. So from 190 to 400, 190 to 600, or 190 out to about 1 micron, depending on the needs and what the system is going to be used in. So from those absorption spectra, we can create this quantification model that now relates the absorption units to a concentration level, and you can see here that it's a fairly linear response over a fairly broad range between just more than around single-digit ppm up to 500 ppm, and then applying this model. Now on the left, when we're just looking at the measured absorbance we're just looking at the broad band of, in this particular case, there was a 275 nanometer absorption band that we can then correlate that so that we can predict concentration. And in this case, there would be two uses of there's the cleaning validation, which is measuring the elimination of product, but it's also a real-time cleaning monitor or cleaning status indicator. So in this case, you can see that in this plot on the right, as you start rinsing your vessel, you start seeing that the solvent starts picking up the API, and eventually after a few minutes, it's saturated with the API around a little over 100 ppm, and so further rinsing of this product really doesn't accomplish anything. So with this indicator, this would also just be a valuable real-time feedback, so that now the vessel can be purged and rinsed with clean solvent until we would see the opposite curve of this, where it's finally the elimination of this product, whether that's down to a 1/1000th or to a ppm standard. And finally, wrapping this up, what all of this actually means as a metric. One of our customers was able to provide us with the dollar amount, and in this case, after the use of a single UV cleaning installation, you can see here a $4 million per year savings, and that's in solvent alone. This number came before this last year or so of drought conditions that have put perhaps an even higher prize on water conservation. So certainly the ROI on a solution like this is quite rapid. So finally, summarizing what we've talked about, that the use of a direct inline solution like a direct UV spectroscopy for cleaning validation certainly it conserves time and money, and now very importantly it conserves water as well. These solutions between Prozess and with Helma are certainly FDA, USP, GMP compliant, 3A certified. And Helma has the first and currently the only 3A certified probe. And like I just said, with metrics like the ones that I had shown before, in the 4 million savings per year with a single installation, that shows a very quick return on investment. So with that, we thank you all for listening, and look forward to answering some of these questions.
Great. Thank you, Chad and Evan. It is indeed time for questions from the audience. Just as a reminder to the audience, if you'd like to submit a question, you can do that by typing it in the Q&A box, and you can find that by clicking on the red Q&A widget at the bottom of your window. Okay, first question. What are the detection limits of the UV solution?
Sure, I can answer that, and that's generally dependent on both the API or what you're cleaning, as well as the solvent that you're using to clean that. But typically the low range of that will be in the low parts per million. and can occasionally be fractional parts per million as well.
And what is the spectral range of the Prozess system? And what is the resolution and spectrometer used?
Sure, and I had mentioned that briefly. We have a number of UV spectrometers that we'll use depending on what the other purposes of the device will be. But all of them start at 190 nanometers and will extend longer from there. So we have a 190 to 400 option that provides fractional nanometer resolution. We have a 190 to 600 nanometer option, as well as a 190 up to around 1 micron solution, where certainly the range and resolution trade off.
Excellent. Does the manufacturing equipment have to have a drain or bypass line for this method to be able to be used?
Not necessarily, and perhaps I can let Evan field that on the use of these insertion probes as well.
So, of course, the way we've described it, the drain line would be a good spot to monitor the concentrations in the effluent, but the probes can absolutely be designed with sufficient length to be inserted from the top or the side into a reactor and monitor the cleaning there. So as you cycle through your cleaning process, you can continuously monitor the concentrations and breakdown of the key components that you're looking at.
Okay, very good. This, I think, was in reference to a specific slide shown. It says the concentration of API measured should decrease with time. Why the increase?
Sure, and there are two reasons for that. Or actually, sorry, there is a reason for that, and let me. I think we'll talk about this slide in general. I think this is what's being questioned, and as I had explained here, this is a case where the solvent actually got saturated. Eventually, with rinsing with the solvent, it can rinse enough residual API, and then it must be drained after it's saturated. So, yes, you would see this as kind of a cleaning monitor approach, so that now there's no need for excessive rinsing with a saturated solvent, so you could drain and refill until then you would see the elimination of the product, which would be the downward curve.
Excellent. Have you gotten any regulatory approval of your UV-Vis cleaning method?
Our systems definitely are the 21 CFR compliant, GMP compliant. But then typically the cleaning method itself is done by the end customers. It's up to their particular protocols to correlate the direct UV approach with their existing approaches. That is typically a multi-step process of using the two in tandem to show correlation before ultimately relying on an inline approach. And again, that would be up to the customer.
Okay. Do you have to identify all the potential components of the rinsate?
No, not necessarily. So just from a broad scale application, because the solvent is typically UV transparent, having some UV absorption is indicative of having some cleaning process still occurring. Ideally, yes, it would be nice to characterize the spectral absorbance of each product or of each component, but typically you would qualify this around the most dangerous or that product which would be which would carry the highest risk in being carried forward into subsequent batches.
Great. So why do you need to use a 3A certified probe even if it isn't required?
Well, I mean, to start, if it isn't required, you don't have to use it. But that being said, the benefits afforded by the hygienic design of this probe allow you to be certain that as you run your cleaning cycles, that you're not actually depositing your ingredients onto the probe itself. The probe is available with essentially the same design without the 3A certification for a reasonably lower price. Additionally, we have a wide range of other probes that can be used. So if you're not concerned about biologics growing, there are certainly other options.
Okay. How do you correlate the concentration in the effluent to the residue on the surfaces of the equipment being cleaned?
That's tricky. It's difficult. So these would be you can empirically, as I had talked about before, that the process of qualifying and validating the UV approach would be multi-step, and I have seen some attempts at correlating the direct UV spectroscopic approach with the swab tests, but this measures what has been picked up in the rinse aid.
Okay. What is the spectral sampling rate for the detectors?
Typically 1 millisecond or 1 kilohertz measurement times is kind of the fastest. That not just the sampling rate of the detectors, but how quickly we can process and output the data. It's typically around 1 millisecond or longer. Very few installations are actually being measured that quickly. They'll typically average over some period of a second or multiple seconds.
Okay.
This averaging method can also be used to accommodate processes where you have bubbles in the process itself or in the effluent during the cleaning process. The bubbles will only be in the light path of the probe for a brief period of time, and by averaging, you can essentially eliminate the aberrant data from the overall results.
Okay. How stable is using a spectroscopy device in a CIP environment?
I think a lot of that comes down to the sample interface. So I'll jump on the end of Evan's answer there. That with doing things like bubble traps and maintaining flow rates and temperatures that can stabilize your spectra, and Evan, I think a lot of that is also in the actual sample interface. And ensuring consistency.
Certainly. I mean, the sample interface itself is very rugged and robust and has been built for consistent, stable readings. So all of the optical components have been tested for the effects of pressure and temperature. They are fixed, and over time you will see very little change due to the sample interface itself. Additionally, the 3A probe has been designed with a leak detection feature that if for whatever reason the seals on the probe have failed and something would start influencing your data like that, it would drop out and you would know immediately.
Excellent. Maybe we'll just do one or two more questions and then wrap up. So you had mentioned something about bubbles. This person says, we don't always have a clear stream to measure. Can your solution be used with bubbles?
So I believe what I said before is actually potentially the answer to this, that bubbles are not necessarily a real problem because they typically move very rapidly out of the light path. So the orientation of the probe to the stream and the bubbles themselves can be placed to eliminate that. Additionally, we have a specially designed sample interface with a vertical slit whose geometry is actually specifically designed to exclude bubbles from the light path.
Excellent. Here's one more question. This person says, to me this seems like a great way to determine the length of rinses and circulation times. But can you actually use this to confirm that all of the equipment is cleaned? For example, top ports on tanks that might not be properly covered by spray balls?
So this is dependent on the rinsate. So for those components that are not picked up by the rinsate, this would not be a solution. So typically that would be something evaluated in the qualification and the validation of the approach. Going when you're first demonstrating this approach versus your accepted protocols of swabbing or HPLC. That would be determined at that point, and if you did end up with sticking points that would not be picked up in the effluent, then it certainly wouldn't proceed to a validated approach.
That being said, I believe that using a diffuse reflectance probe can actually you can look at specific areas with somebody handling the probe as opposed to swabbing and sending off to a lab and waiting for results.
Excellent. All right, with that, I think we'll wrap up and we can answer other questions afterward offline. Well, thank you both for that informative presentation. I'd also like to thank all of you in the audience for participating today and for asking all of those interesting questions. And I'd like to thank our sponsors, Hellma Analytics and Prozess Technologie, for making today's webcast possible. This webcast will be available for on-demand viewing through August of next year. You will receive an email from Spectroscopy alerting you when the webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.